Hello, welcome to Cancer Research Demystified. It's still the coronavirus crisis, so I'm PPE'd up to the max and I'm on my own in the lab, and there's no Susan, I'm afraid. So I'm just going to do a really quick video on what we do for Christmas. So some scientists work all through Christmas, but some of us decide to take a break and visit family. That's going to be harder this year, but I still want to take a break, so what I'm going to do is put my cancer cells to sleep while I'm away, so I don't need to look after them, and wake them up when I come back. We have other videos on cancer cells, what we do with them, what they are, how we get them, and I'll link to those in the links. But for now, I'm just going to show you what we do to put them to sleep for a little while so we can take a break. So the cells live in an incubator like this. It's 37 degrees C, so it's nice and warm like your body. It's humid, so it's nice and moist like your body. And there's a high CO2 concentration, 5%, which is higher than the air around and a bit more similar to what you get in your body. So they live in here. And they live in plastic dishes. And these particular cells grow on the bottom under this media, which is a bit like squash or cordial. It's full of salts and sugars and all the nutrients they need to survive. And sometimes you can see a fine little dust. They're a bit small, they're tiny, so you probably can't see them, but I'll show you what they look like down the microscope. So this is me down the other end of the lab, and this is our microscope. I've put my cells on here, and we can have a look to see if they're ready for freezing. So the cells look healthy. There's a few dead ones, but that's relatively normal with this cell line. They don't look too many of them. There's not too few of them. They look happy and healthy, so they're going to be ready to be frozen. So we work in a hood like this, which keeps the air inside this hood sterile. And so we only expose the cells when they're outside the flask in a sterile environment. They can get infections like you can. And we don't want to give them an infection before we freeze them, because when we wake them up, they'll die very quickly. So I'm going to freeze 4 flasks. And I freeze them in freezing media. So this is essentially just tons and tons of nutrients to keep them happy, as well as some DMSO, and that's a chemical I'll explain in a bit. So the cells have been removed from their flask and they've been put in little tubes like this. We put them into an isopropanol bath. And then this makes sure that the cells freeze very, very slowly. As I said before, there's also a chemical called DMSO mixed in with these cells. Both of those chemicals and the slow freezing are designed to make sure that we don't get ice crystals in our cells. The reason for that is if you imagine you put a can of Coke or a can of beer in the freezer just to cool it down, you forget about it, you come back, it's just blown up, it's exploded. So that's because ice expands, and if that happened inside our cells, they'd burst open, would try and wake them up from their sleep, and they wouldn't be alive. So we cool them very slowly using the isopropanol bath, and we use a cryoprotectant to help stop those ice crystals forming. So we've got our cells now. We're going to put them into one of the freezers, and it slowly reduces the temperature 1 degree every minute because of that isopropanol bath. What we want when it's cold is the cells are asleep. There's no reactions going on. They don't need anything. There's no chemical or biological reactions. It's just too cold for anything to happen. And the cells just stay hibernated like that until we want to wake them up. So that's -80, that's the first step. Once it gets to -80, I then put them in liquid nitrogen, which is even colder. So that's it. Merry Christmas, sleep well cells, and I'll see you when I'm back after the holidays.